Pajamas. Alright, so today we're going to talk about chromosomes, or as I like to sometimes call them, the chromosomes. Now we know that chromosomes are large sections of our DNA that have been separated out. Specifically, we really only see chromosomes during cell division, so mitosis or meiosis. Most of the time, our DNA is in a form called chromatin, which is a super condensed form of our genetic material, so that all of it is together, super condensed down. Kind of think of it like a ball of a thread. You have a really, really long piece of thread, but you ball it up and wrap it around something. It's nice, small, compact, and it can fit in a small space. Chromosomes, though, are when we chunk that chromatin into specific portions, and that's really what our chromosomes are. So it's still coiled up DNA, it's just that it's been chunked at specific locations. And humans, we have 46 total chromosomes. It comes in 23 pairs. So our diploid number, diploid meaning the full set of pairs, is we have a diploid number of 2n, or 2 times 23, because we have 23 pairs, so a total chromosome number of 46. Now, where do we get our pairs from? Well, that's very simple. I'm glad you asked. We get one from mom, one from dad. We've mentioned, there's another video that we've done on sexual reproduction, where we talk about how we make these special cells called gametes. They're haploid. They only have one of each chromosome, so that you have one of each chromosome from mom, one of each chromosome from dad. They come together. You now have pairs of chromosomes. Now, let's talk a little bit about chromosomes, like their structure and all that. So, a chromosome is generally, if you just want to think about it, it's just a section of chunked up portion of DNA. Now, a lot of times, though, we'll draw it like this, because in that case, this is showing that it's gone through DNA replication, and these are the sister chromatids. Well, you might notice a lot of times we draw that little tiny thing right there. That's called the centromere. The centromere is a region with proteins that holds the sister chromatids together until they're ready to be separated during anaphase. So that holds the sister chromatids together. We also have these things called, I left my rag over there. Silly Komar. Well, luckily for me, I got tissues. Alright, so we also have these things called we talked about centromeres. We also have these things called telomeres. So I'm going to draw these a little bit better. So telomeres, I know I said I was going to draw it bad, but look how fat that side is. Anyways, telomeres are the ends of the chromosomes. Now, these are actually very important. When your DNA goes through DNA replication, and you mentioned how it's separated out, and you have polymerase going down, reading the template strand and adding the associated complementary bases as it goes. Now, once it gets to this point here, so it's already built all this part here, it's still hanging on to there. It can't build that section. So what happens is that last little piece does not get duplicated. So when it gets to the end of the chromosome, essentially it does not duplicate the very, very end. So we have, I'm going to draw this a little bit better. A, T, C, C, G, C, A, T, A, G, C, G, A, T, C. Well, the DNA polymerase maybe it's located right here. So that's the polymerase. DNA polymerase. And it's already gone through, it's built the new strand right here. So T, A, G, G, C, G, T, T, A, T, C. But it kind of builds as it goes. Well, there's nowhere else for it to go here, so it can't build that last section. So it just detaches. And we're left with a portion that cannot be built. So what happens is, over time, as your cell goes through its cellular life cycle and it goes through divisions, the telomeres get shorter and shorter and shorter. Once there are no more telomeres, then the cell can no longer divide. It finishes up its lifespan, it goes to apoptosis, which is programmed cell death. And again, the reason for this primarily being because, I'm trying to avoid that glare, I'm sorry. The reason for this primarily being because you don't want to lose any essential genes. So these telomeres, they're made up of these long repeating sequences. And the point of them, or what we believe is one of the purposes of them, of these long repeating sequences at the end of the chromosomes, is to provide a buffer. So that when we can't build that last bit, instead of losing a part of a gene, we just lose some of these repeating sequences. This way, all the genes are still intact and they can still be expressed normally. So those are what we call telomeres, the ends of the chromosomes, and they do tend to shorten over time. Now, we mentioned certain terms. Our cells, the vast majority of them, are what we call diploid, meaning we have two of each chromosome. And so we have 23 different types of chromosomes. 2 times 23 in humans, that number is 46. Our gametes are haploid, meaning that they just have one of each chromosome. So again, we have 23 types, one of each would be 23. But there are some disorders and some organisms where, it's not just like this. What I mean by that, in terms of other organisms, we have this idea called polyploidy. So polyploidy is when you have extra whole sets of chromosomes. So instead of being diploid, if you're a polyploid, you're going to have a different number in front of that N. So let's say humans were polyploidy and we had 3 of each chromosome. And that means we would be 3N, or 3 times 23, which is 69? Yeah, 69 chromosomes. So it's not just having an extra 1 or 2, it's actually having an extra set, or even more. I believe strawberries I know are polyploidy. I just can't remember what their number is, but they are polyploidy. They don't have chromosomes in pairs, they have them in these larger sets. So we know that our chromosomes typically, like we said, they're in pairs. Homologous pairs. But if you were polyploidy, all of your chromosomes would be maybe you have 3 of each chromosome, or 7 of each chromosome. But the point is, when it's polyploidy, it is a full extra set, or more than one set, but you have extra sets of chromosomes. That's different than aneuploidy. So with Aneuploidy, that's when you might have one extra of a particular chromosome, or you might be missing one of a particular chromosome. So it's not every chromosome has an extra version, but for instance, maybe someone has 47 chromosomes because they have one extra. So 47 chromosomes, all of them are in pairs. With the exception of the 21st chromosome. That one, there are 3 copies of it. So you have everything in pairs, 46 total, plus an extra 21st chromosome. Now that's an example of aneuploidy because you have one extra, and specifically we call this trisomy 21, better known as Down syndrome. There are other cases where you might have 45 chromosomes. Maybe all your other chromosomes come in pairs, but except your your sex chromosomes. Maybe you just have an X and nothing else. So you don't have 2 X's, you don't have an X and a Y, you just simply have an X. And let's see, I can't remember which one that one's called, so give me a moment. And that one is Turner syndrome. Now, what they've noticed is that, or first of all, when you are missing a chromosome like that, where you only have one instead of a pair, they call that monosomy. Mono meaning one, and then somy referring to the chromosomes. You only have one of that chromosome. Now, monosomies are very rare. If you have one version of most of the chromosomes, like most of your autosomes, generally speaking, it's very hard to survive that because our cells are programmed to read 2 chromosomes. So generally, that doesn't have a very, very high survival rate. Although what they have noticed is that a lot of the aneuploidies, if they're of the sex chromosomes rather than the autosomes, there's a much higher chance for survival. So we mentioned monosomy of the X chromosome, which is Turner syndrome. There's XXY, so trisomy with 2 X's and a Y, that leads to Klinefelter syndrome. If you have 3 X's, we call it triplo X. XYY, so 1 X and 2 Y's, we call that Jacob syndrome. And there's any number of other combinations. So those are trisomy of the 23rd pair, or the sex chromosomes, with the exception of Turner syndrome, which is monosomy of the sex chromosomes. There are also, we mentioned trisomy 21. That is one of the more common trisomies or aneuploidies of an autosome. Sometimes trisomy 22 can also lead to Down syndrome. The only other 2 that are really common are trisomy 18 and trisomy 13. Now, those tend to have a lot more severe effects than Down syndrome. And let's see, pretty much any other trisomies generally result in spontaneous abortions, which are miscarriages at different stages of development. Same with monosomies. Most monosomies will result in spontaneous abortion. And even polyploidies, in humans, we are not programmed, our cells are not programmed to read more than that pair. So when you have a complete extra of every single chromosome, the cells freak out and it leads to a spontaneous abortion of the developing fetus. But yeah, I think those are the main examples I want to talk about in terms of, chromosomes and all that. So I hope that helps you guys understand these a little bit better. And until next time, you guys take care and stay healthy.